Hello everyone, immunity part 2. Today we're going to talk about B lymphocytes. B lymphocytes are part of what we call humoral immunity. So basically it's called this because antibodies, which are produced by B lymphocytes, circulate in the blood and tissue fluid, which used to be called the humour, or the fluids of the body. So B lymphocytes produce antibodies, they circulate in the blood and the tissue fluid, and therefore it's called humoral immunity. This follows on from the cell-mediated immunity that we looked at last lesson, which was the T lymphocytes. And if you remember, ultimately we ended up with lymphocytes that respond to antigens presented on a body cell, so for example a phagocyte or a host cell, and you ended up with 3 different types: T cytotoxic, T helper and T memory cells. The T helper cells are the ones that are involved in what we're going to talk about today, because they stimulate the B lymphocytes to divide. So just kind of remember that when we look at B lymphocytes. B lymphocytes respond to any antigen that's just kind of kicking around. So here's our evil pathogen. So that's our pathogen with an antigen on its surface. B lymphocytes also respond to antigens that might be on, for example, toxins. So when pathogens produce toxins, B lymphocytes will also respond to those. So the first thing that happens is what we call clonal selection, which, as the name suggests, is the selecting of a complementary B lymphocyte. So you've got a B lymphocyte with so these are our B lymphocytes, and they have got antibodies. They've got antibodies on them, and so they also have, let's not worry about the receptors. So B lymphocytes have got antibodies on them that are complementary to the antigen, so same as, and there are a variety of B lymphocytes. They're all different, same as the T lymphocytes. There are literally millions of them, so any kind of pathogen can be responded to. So the complementary B lymphocyte is selected, which in this case is this one. It binds to the pathogen and ultimately ends up displaying the antigen on its surface. So this is something a little bit different. So the B cell is almost like a phagocyte in that it processes the pathogen, takes the antigen, displays it on its surface. Okay, so that's whichever B cell has the complementary antibody to the antigen will grab the antigen, ultimately process it and display it on its surface. Now, what type of cells respond to displayed antigens? That's the T lymphocytes. So the complementary T lymphocyte will come along and bind, and so those two will bind together. These are the T helper lymphocytes. So the complementary T helper lymphocyte will come and bind to the antigen, and that's being presented by the B cell, and that will cause a huge number of that B lymphocyte to be cloned. Okay, and the process by which these are cloned is mitosis. So cloned by mitosis, and that's like a key phrase to stick in an answer about this. Okay, so just to recap, B lymphocytes all have different antibodies on them, and there are many, many, many of them. If they find an antigen that is complementary to their antibody, they will bind to it. They'll process the antigen and display it on their surface. T helper cells with the complementary receptor will then bind to the B cells and cause those B cells to clone themselves by mitosis, so we have very many of them. That you've then got, like last time, you then got the process of differentiation. And differentiation basically involves B lymphocytes turning into two different versions. Version number 1 are plasma cells. Plasma cells are the ones that do the useful stuff, basically. So plasma cells produce antibodies. And ultimately, we'll talk way more about antibodies next lesson, but ultimately, antibodies are what actually destroys the pathogen. So there are the antibodies. They will then go up here and destroy the pathogen. So that's one type of cell produced by differentiation. And then the other type of cell are B memory cells. And those are the ones that are involved in immunity. So they stay in the blood for a really, really long time. And if you then get that same pathogen entering the body again, these B memory cells will reproduce themselves very, very quickly, differentiate into plasma cells very, very quickly, produce antibodies very, very quickly, huge numbers of them, and overcome that pathogen before it can make you feel ill.